All right, so today's lecture is going to be on DNA technology and genetic engineering. Of course, a lot of people might ask, why do we care? Why do we care about genetic engineering? Why do we care about DNA technology? I mean, isn't that something that we hear about in all these movies where it always goes wrong? Somebody uses genetic engineering to make some dinosaurs, and then they turn around and eat everybody. So is this something we really want to risk? Is this something we really want to do? Well, I am glad you asked. And so today we are going to be learning all about genetic engineering and DNA technology and why modern biotechnology is so important to modern civilization. So here's our critical thinking question. The year is 2170. You learn that a member of the Enterprise crew, Dr. Flox, will be a guest speaker at RIT. Dr. Flox will be presenting research in which he claims to have developed a vaccine that can prevent 99.4% of cancers in humans. Your friend says that she does not think you should go to the talk. She says that Dr. Flox's research is obviously biased. The fungus that is used as a main component of the vaccine is grown on a farm owned by the Flox family. And with this new vaccine patent, Dr. Flox stands to gain unparalleled profits. So, this opens up the question, what do you think about this? Is Dr. Flox biased? Can his research be trusted? Can we really listen to this man knowing that he is going to gain so much money from the profits from this new vaccine? And given all that we know, can we really trust this product that he's trying to give to us? But it gets even better. Your friend also cautions that Dr. Flox is from a species that openly practices genetic engineering on humanoids, counter to the laws that were put in place on Earth after World War III. She asks if we can really trust someone from a culture that embraces genetic engineering. Furthermore, Dr. Flox is part of an obscure religious order on Denobula that many regard as a cult. Not to mention Dr. Flox's belief in the Denobula Liberation Movement, an obscure political movement that is considered radical even by many people on Denobula. So this opens a lot of questions. Given his obscure and fringe religious and political beliefs, along with being from a culture that practices genetic engineering, can we really trust him? I mean, after all, here on Earth, we had the Second American Civil War, we had World War III, and we had the eugenics wars. And of course, it's not in that order, but we saw so many horrors take place on this planet, and it was not that long ago. So, I mean, given that this man is from a culture that wants us to embrace genetic engineering, can we really trust him? Can we really trust that this is going to be good for us and for our children? Can we really trust that his science is sound and kosher and that it's going to be good for us? So this brings all of these questions back around. Since Dr. Flox has so many radical beliefs about politics, religion, and ideology, and since he stands to gain such a great profit, made off the backs of humans, by the way, on multiple fronts, can he really be trusted to be honest with his research? I mean, can we really trust this outsider? I mean, and don't get me wrong, I'm not against aliens. I'm not against people from other worlds coming to Earth and speaking at RIT, but we do really have to question, I mean, how much can we really trust this guy given that his culture's ethical stances on technology such as genetic engineering just does not match with what we have decided as the human race here on Earth. So, and also, while clinical trials are still early, many scientists have questioned these claims, including those at Cambridge, Harvard, and the Vulcan Science Institute. So, whether or not you think this is something we can trust, whether or not you think this is science that we really want to risk, um, well, I think these are some questions we should really be asking. All right, so now this brings this whole critical thinking question back around. So, what is an ad hominem? You'll notice that in this scenario, nothing was brought up about Dr. Flox's actual science. I mean, it was mentioned that there's a few scientists at other well-respected institutions that were skeptical of his work, but scientists are supposed to be skeptical. We're supposed to be open-minded skeptics. I think that's the healthy way to do science is you're not skeptical to the point of personal incredulity, um, but you're also not so open-minded that your brain falls out. So this question here, what is an ad hominem? An ad hominem is a logical fallacy where you attack the person rather than their argument. So in the critical thinking scenario, the question is, well, can we really trust Dr. Flox given his beliefs about politics and religion, and his culture's view on ethics? So it was an attack on his character, on his race, he's denobulan rather than human, on his political beliefs, on his religious beliefs. So there was a lot here where his claims are being called into question, but none of it had anything to do with his science. The proper response to something like this might be, hey, we have some skepticism given all of this, which maybe would be fair. However, it should really come down to looking at the data and saying, does his data hold up? What do the clinical trials show? And maybe at that point say, hey, we want to have the clinical trials done by a third party. So basically, a lot of what I would urge people to learn from this fictional lesson is don't just dismiss the arguments of somebody based on ad hominem attacks. Don't dismiss it just based on, oh, well, he has political or religious beliefs that I don't agree with, or he's from, this particular cultural group. Things like that don't have any bearing on the logic of the person's argument or on the legitimacy of their science. And this also opens a few more questions. Should we hear the perspectives of scientists from different cultures from our own? Why or why not? Now, I ask this question as a question that should be obvious. We should hear different perspectives from our own. That's part of why we have universities and why we have academia, so that we can hear different perspectives. And should we hear from those who we disagree with in terms of politics, religion, or ideology? Why or why not? And I think that's something that you can really decide for yourself. I think most people will say yes to this in theory, but then the question is, are you going out of your way to hear the arguments of scientists and academics who maybe hold to a different view from your own? Now, of course, the scenario is fan fiction. This is based on Dr. Flox, who is a character from the Star Trek franchise, and he's a little known character from the franchise, but he's one of my favorites. Um, and of course, this is with a lot of fan fiction and creative liberties for the sake of education. So, fan fiction is allowed for the sake of commentary, for the sake of education, and of course, I'm not making any money off of this. So, Dr. Flox, I really admire this particular character because he's from a culture that really embraced biotechnology. So the vision of the future that is presented in Star Trek Enterprise, which takes place 100 years before the original show, it imagines a future where humans are very afraid of genetic engineering because we had some conflicts. We had the Second American Civil War, the eugenics wars, and World War III. So we had a very bad, very negative history with this particular technology, similar to how we might have seen nuclear power just after World War II. However, I would ask the question, is fire good or is fire evil? And I asked this to one of my students, and she said, well, it depends on how you use it. Are you using it to burn somebody's house down, or are you using it to warm someone's home? And so that's really the same with, I would say, nuclear power, genetic engineering, and even fire. I don't think technology is inherently evil. In fact, I will say I think the technology is inherently good, but we can use it for evil. And I think the same applies to genetic engineering technology. On Dr. Flox's fictional world of Denobula, they've used genetic engineering to heal, to help make people whole, to help make their world a better place. Whereas on Earth, we used it for war and to create dictators in the fictional Star Trek universe that ruled over large portions of humanity. So this fictional device really offers us two options. Do we use genetic engineering, which we will be talking about today, for the sake of destruction, or do we use it for the sake of building a beautiful and better future? So now we have some basic terminology. So biotechnology is defined as the manipulation of living organisms or their components to produce useful products. Now, biotechnology in some sense has been around since ancient times. People used microbes to make wine, cheese, beer, soy sauce, you name it. However, modern biotechnology really began in the 1970s when a company took the human gene for insulin and put it inside of bacteria, after manipulating it a little bit, of course, so that they could make human insulin. So recombinant DNA technology, which would fall under this umbrella as well, is applied science that explores applications of cutting, splicing, and creating DNA. So we're taking pieces of DNA, chopping them up, sewing them back together, and saying, okay, what can we do in terms of manipulating DNA in order to create new products? And genetic engineering is we are manipulating the genetic makeup of cells um, and using them to alter organisms. So, genetic engineering, I was once asked, is selective breeding a form of genetic engineering? And I would say no, because it's not. Genetic engineering, I would argue, is you are taking DNA and changing it directly. Whereas selective breeding is you are just taking the genetic variation that is already there and selecting from that and choosing which organisms, such as which animals or crop plants, you're going to breed. So these are some basic terms here, and if you are interested after this lecture in learning more about this topic, RIT actually offers a full major in biotechnology. So I would definitely love to chat if you would like to learn more after we're totally done with the lecture today. So DNA sequencing is obviously very important for biotechnology. What do we mean by DNA sequencing, first of all? So DNA sequencing is you are determining the sequence of letters in the DNA. So I might use the term letters, I might use the term nucleotides, I might use the term base pairs, but this basically means the same thing. So the series of A's, C's, G's, and T's that you find in DNA, we determine that using sequencing. Sanger dideoxy chain termination is the classical method of sequencing DNA. It was used for many years and it still is. And modifications of the Sanger method were used to automate and enable the sequencing of the Human Genome Project, which was completed in 2001. So I'm going to try to see if I can draw some pictures here to help give you an idea of how this works. Let's see, do I have... Oh, there we go. Okay, there's the little cursor. So if we have a piece of DNA like this and it ends in an A, do this all Khan Academy style. And so let's say we have, this is 10 base pairs long. And then we have another one. We have 11 base pairs, and that ends in a C. And say we got another one that's, let's do 9 base pairs. Because why not? And this is a T. So 9 base pairs followed by a T, then 10 followed by an A, then 11 followed by a C. What we can do is we could say, well, okay, you have 9, and then the 10th one is a T. And then you have 10, and the next one is an A, and 11, and the next one is a C. So T is the 10th one, A is the 11th, C is the 12th. So we know that this would be T, A, because the A would come after the T, and then the C would follow. So we have 9 base pairs, and then we have TAC. And then we would gradually go in and figure out which base pairs come before this and which come after. So if we have one that's, say, 8 base pairs long, and then there's an A. Well, okay, so we know the 9th one is an A, the 10th one is a T, 11 is 12, or 11 is A, I'm sorry, and then 12 is C. So A, T, A, C. And so it basically goes through like this, and it figures out which nucleotides come next in the sequence. And by putting that whole thing together, you can figure out what the sequence is for your fragment of DNA. So initially, the Sanger method used radioactive dideoxy labels. So these are dideoxynucleotides, meaning that as the DNA chain grows with the polymerase, when the dideoxy comes in, it is going to terminate the growth of that DNA. So once you have a dideoxynucleotide introduced, it can't keep growing past that. So initially, these dideoxynucleotides were radioactively labeled, and we had to have 4 lanes. So you had to have A, T, G, and C, each in 4 different lanes, because you couldn't tell the difference between the radioactive labels. So you had to have 4 different lanes. Now, somebody very smart came along and said, well, let's try labeling these with 4 different colors instead of having them be radioactively labeled. So as you can see here, we have T labeled as red, C labeled as blue, A labeled as green, and G labeled as purple. So let's see if we can do this here. So right here, we can see that this 3' N ends in a purple, so that is G. Then there's another purple, so that's another G. Then there's a red, so that means that's a T. And then right here, there's a blue, so that means it's a C, followed by A, T, A, G, C. So by putting this together, you can say there's this nucleotide followed by this one. So G, G, T, C, A, T, A, G, and C. So that gives you your sequence right there. And by having these different colors, this means you can do the sequencing all within a single gel. So instead of having 4 lanes within a gel, you can have 1 lane. And this also helps the process of automating this and making it where this can be done a lot more efficiently. So you can see right here how we have different computers and instruments to read the gel and determine what the sequence is. This is a lot better than having a lab tech have to do this all by hand. So returning to this slide, what is the purpose of all this? We want to know what the sequence of A's, C's, G's, and T's are in our DNA sample. So the Sanger method, of course, was standard. It was used for many years, and it enabled us to complete the sequencing of the Human Genome Project. So this is still the gold standard method. It is still very widely used. However, there are other methods that have arisen in the years since Sanger was originally invented. However, like I said, Sanger is still a good method. It is still the gold standard. It is still the most accurate if we are looking at a piece of DNA of the appropriate size. So after the Sanger method was used for quite a few years, eventually we developed something called next generation sequencing. And of course, this is an automated method. This is also known as deep sequencing, massively parallel sequencing, or high throughput sequencing. So if you hear those different terms, this is what it's referring to. Now, next generation sequencing, there's plenty of videos on YouTube you can watch if you want to know how it works. However, at this level, I can definitely tell you it's a very confusing process. The most important thing about it is it takes many pieces of DNA and sequences them all at once. So you don't have to just sequence one fragment of DNA at a time. You can sequence a whole bunch of them simultaneously, and that's a huge advantage of next generation sequencing, as we will see here shortly. So the way this works is we're taking DNA, cutting it into many fragments, and sequencing the fragments simultaneously. So there's just a whole bunch of fragments, they're being sequenced all at once, and computers are able to analyze the data and sort, align, and assemble the sequence. So what they're doing is basically taking a bunch of pieces of sequenced DNA and then figuring out where they overlap with one another. And by figuring out where they overlap with one another, they could say, hey, well, this piece overlaps this piece, so that must represent a continuation of the overall DNA in a living organism. So let's see if I could draw a picture here. So if we have this fragment here, and we figure out that it overlaps with this fragment right here. So here's a fragment. Let's see if I could do the little laser pointer. So here's a fragment right here. Here's a fragment right here. But they overlap right here. So right here is where they overlap. This means that we can figure out that in the original piece of DNA, you had something like this. So by saying this fragment, which we have in our sample, and this fragment, which we have in our sample, since they overlap so much, we figure they must have originally been part of one piece of DNA, and we can put them together like this. Now we might also have it where here's another fragment. Here, let's use a different color. Let's use purple. So this fragment overlaps right here. Okay, cool. So it means we can figure out that the original DNA sequence kept going. And I know I'm about to color my head here for a minute. All right, so there we go. So we have this fragment, fragment 1, 2, and 3, and by finding where they overlap and putting the whole thing together, you can figure out what the original piece of DNA looked like, and this is the power of next generation sequencing. So here we have an individual who wants to have her DNA sequence. Maybe she is a patient and maybe she has some kind of illness, or maybe she's a student, or maybe she's just curious about her DNA. So what we can do is we can take her DNA, so we take her chromosomes or her entire genome, and we break these up into small fragments. Let's assume that we want to sequence her whole genome just for the sake of illustration. So these chromosomes are cut with restriction enzymes, producing fragments of DNA. So restriction enzymes come along, they kind of look like a little Pac-Man. So let's go ahead and draw that right here. So why not? We got a little Pac-Man type enzyme going on. So this is going to come to the DNA, and it is going to cut the DNA at a particular location. So it's going to come here, cut here. If it finds a particular sequence, then that's where it will cut. And so we have all these little fragments here, and then we have fragments that are several hundred nucleotides in length. They're sequenced at the same time, so they're sequenced simultaneously using next generation automatic sequencers. And then computers take these fragments, figure out where they overlap. Let me use a little laser pointer here. So we figure out where they overlap, and yes, this is a little oversimplified because we want more than 2 or 3 nucleotides in the overlap, so the picture here I think is a little too oversimplified. And then we're able to put the whole thing together. And then you see a technician right here working at an instrument, figuring out what Claudia's DNA looked like originally. Now here's where things get really interesting. We are now on to third generation sequencing. So third generation sequencing, also known as long read sequencing takes very long pieces of DNA and it's able to sequence the whole thing. It's also able to tell you if it's methylated or non-methylated. So the first time I saw a third generation sequencer, I didn't believe it. I was watching a documentary and there was a scientist and she was taking samples from volcanic hot springs and sequencing DNA from those samples. So she was probably looking at microbial archaea that lived in those hot springs. And there was just this tiny little device. It was like this big, like the size of an old MP3 player. And they said in the documentary, oh, she's using this device to sequence the DNA. And I said, no, that can't be right. That can't be true. That can't be what's going on. The narrator must be wrong. He must have misunderstood something, or somebody messed up on the script. Like that can't be what's happening. But actually, it was what was happening. She was using a tiny little nanopore sequencer to sequence the genomes of the archaea that she was studying. So the Human Genome Project was first completed in 2001. Now, this didn't mean that we knew every single nucleotide of the human genome. There was still a little bit of question at this point. So this was a huge project, took 13 years and cost $2.7 billion. Today, using the newer methods, we can do the same thing for under $1,000. Now, during this time, there was a bit of an arms race between Francis Collins and Craig Venter. Francis Collins was the head of the NIH, and he was trying to complete the Human Genome Project, and at the time, this was seen as being on par with the moon landing. This was a huge project, a lot of labs, and a lot of people were involved in this. But Craig Venter, being a big industrialist, came along and said, you know what? I think I can do it faster, more cheaply, and do it better than you can. So shots were fired. Now, initially, when I had learned about this, Francis Collins, who was the head of the NIH, was depicted as being very, very much the underdog in this. So he had, limited federal funds, and he was trying to get this project done. Whereas you had this big industrialist come along, who is also trying to sequence the human genome. And so Francis Collins was very much depicted as being the underdog. But looking back on the situation and learning a little bit more about the world now that I'm a little older, I actually think it's very honorable that somebody like Craig Venter spent his own money and was taking on a lot of risk in order to sequence the human genome, like I said, using his own money and using his own company to try to do this. So there was basically this space race. It was basically the equivalent of the space race, but it was a human genome race to complete this human genome, and it was a huge competition between these two gentlemen. Now, ultimately what happened is the Human Genome Project was completed. Both parties were involved in this, and both men were honored by President Clinton at the same time and thanked for their contribution to sequencing the human genome. So it's definitely an interesting part of history if you end up wanting to look further into it. So here is a small device used for this third generation sequencing technology. So here we have the cost per genome in dollars. So initially it's very, very expensive to sequence a genome. And over time, this became cheaper and cheaper and cheaper. And it dipped to below $1,000 right as we're approaching 2019. So when I was an undergraduate student earning my associates back here in 2011, I read a book by Craig Venter called The Language of Life. And in it, he was arguing for what was going to happen once it costs less than $1,000 to sequence a full human genome. And I presented on that, talked about it, and at the time I said, well, this is maybe going to be controversial for me to say, but I'll bet that once we can sequence the full genome, we could even take our phones and look at the results on our phones and see what types of diseases we might have risks for, see what our ancestry is, and get a lot of other information. And of course, as people understand the human genome more and more, once we have our own genome sequenced, we can retroactively apply that knowledge to our sequences and get updates, learn about other diseases we might have risks for, and learn about other information in terms of our ancestry. And at the time when I was saying this, it felt like fantasy, it felt like science fiction, but now this is basically the standard. If people want to get their full genome sequenced, they expect to be able to look at the results on their phone. So here are some terms to learn in terms of recombinant DNA technology. So we're talking about isolating and cloning genes. But what does that mean? So recombinant DNA technology involves cutting pieces of DNA, splicing them, so putting them back together again, and copying DNA. So we might take so here's a chromosome from one organism, and say there's a gene right here that we want. So maybe this is a human chromosome, and there's a gene right here that makes a particular protein that we want. And now we have a bacterial plasmid, a small piece of DNA found inside of bacteria. So inside of bacteria you have the circular chromosome, but you also have tiny little circular pieces of DNA called plasmids. So we want to take this gene and pop it into this plasmid. And part of how we can do that is we can take restriction enzymes, which will cut DNA at specific sites, often palindromes. And I can explain what palindromes here are in a minute. So let's underline palindromes because that's an important word. So we will take these enzymes that look like little Pac-Man. Let's draw a little Pac-Man type thing here. Okay. And yes, my illustrations look terrible, especially when I'm trying to draw with a mouse. So this guy's going to come in. Maybe he's going to cut right here, and maybe he's going to cut right about here and right about here. And so we can take this human gene and this plasmid, and what we can do is we can make a cut the plasmid, and then we can ligate this human gene inside of this plasmid. So now the bacteria can come along and make the protein that is associated with this gene. Now, if this bacteria has a slightly different genetic code from humans, or if there's, bacteria also will read the DNA a little bit differently, we might have to change this gene around just a little bit in order to make it where the bacteria can read it. So it's almost like changing a file type. So if I have a file type of Microsoft Word, but I want it to be read in a different type of software, I might have to change the formatting of the file around just a little bit. So it's the same thing with this gene here. We might have to change it around just a little bit. It might not be the exact DNA sequence, so that we are able to get that particular protein. And let's say that's insulin. So we cut out a piece of human DNA and put it into a bacterial plasmid. Using restriction enzymes to cut this, and using DNA ligases to join these fragments back together again. And so we've explained what plasmids are, so small circular pieces of DNA to which desired genes can be added and inserted into bacteria for amplification. So we might take this little plasmid and stick it inside of a species of bacteria that it's going to be compatible with. So I also mentioned palindromes. So What are palindromes? Palindromes in normal language are a word that is the same spelled forward and backwards. But when we're talking about palindromes in the context of DNA, the meaning is slightly different. So you might have A, T, C, and on one side of the DNA, and then C, T, A on the other. So you can see how you have A, T, C, but if we were to rotate this around, it would also be A, T, C. So let's go in and complete this. So C pairs with G, T pairs with A pairs with T, C pairs with G, T pairs with A, and A pairs with T. So this is a palindrome here. And I know I have amazing handwriting right now. Again, I'm trying to do this with a mouse, so it is what it is. But the enzyme would come in and it might cut something like this, or it might cut something like this. Oops. Tried to do a straight line there. Or perhaps it cuts something like this. So different restriction enzymes can cut in different ways. And hey, maybe it actually cuts something like this. The point is, different restriction enzymes will cut in different ways, but they will typically recognize a particular sequence. This is often a palindrome, and the restriction enzyme knows to come in and cut here. From the perspective of bacteria, they do this to help protect themselves from viruses. But from the perspective of human beings, we said, hey, this is a cool tool. We can use this for genetic engineering. So here's an example of a short palindrome, AATT, and how the restriction enzyme will come in and cut it. I like my palindrome example better, but this one is definitely drawn a lot more nicely. So what you'll have here is you'll have the two pieces of DNA, and they'll have these ends, and what you can do is another piece of DNA that is also cut using that restriction enzyme can be used to bind to this. Now you might also have a situation where maybe you need to do blunt end ligation, or you need to stick together two pieces of DNA that were not cut by the same restriction enzyme. And of course we have all kinds of ways to mix and match pieces of DNA. But the main thing to remember is that with transgenic DNA, you're taking DNA from different organisms and you're cutting them apart and pasting them together. So right here, we have a description of recombinant DNA technology, and this is basically going to summarize what it is that we've talked about. So we're going to make bacteria that can produce human insulin, which can be used by diabetics. Now, my grandparents who were diabetic, I remember seeing them stick their fingers when I was a kid, and they needed to monitor their glucose. So being able to manage their diabetes, being able to use insulin, this is very, very important. This is very important for people. So process here is we isolated and purified bacterial plasmids, as we described before, and the human insulin gene. So we needed bacterial plasmids that would work for our project, and we needed the human insulin gene. We're going to cut both DNAs with the same restriction enzyme. We're going to mix the cut plasmids and human insulin gene DNA. So we're going to take both of them, put them in the same test tube, and naturally some of those insulin genes are going to bind to the cut ends of the plasmid, because you have that complementary base pairing. Now, ligase is going to come along, it's going to join the insulin gene to the plasmid. So they're going to start to bond together, and then ligase will come in and seal that bond. So introduce insulin plasmids to bacteria, E. coli in this case, and we're going to screen and select those E. coli which successfully took up the insulin producing plasmid. So we need to make sure they have a plasmid, and we need to make sure that particular plasmid is producing the human insulin. So we're going to go through and look at a whole bunch of colonies of E. coli and figure out which ones actually contain which ones are actually making human insulin. And you might say, oh, here's a colony that is doing what we want it to do. And so you can take some bacteria from that colony and you can grow them in a vat. And you don't need a lot to start with, because within a very short time, 1 E. coli can become 2, 2 becomes 4, 4 becomes 8. And E. coli can double within about 20 minutes. So you have 1 E. coli, you put that onto a plate, leave it in the incubator overnight and come back, and there's going to be a colony with billions and billions of E. coli on there. So this was actually the first commercial technology produced by modern biotechnology, and a lot of people's lives are a lot better because of it. So here we see a summary of what we've been talking about so far. So here inside of the E. coli there's a plasmid. We're going to take this plasmid, it has a palindromic sequence, and we are going to cut this palindromic sequence using a restriction enzyme. And right here, we're going to take human DNA and we're going to cut out the gene for human insulin. Now, this does have to be modified properly. It can't use, when we put this inside of bacteria, we have to make sure there's no introns. Also, I did confirm that humans and E. coli do share the same genetic code. So, we take this intron, or we take this gene, we're going to put it inside of this cut E. coli plasmid, and we are going to ligate the gene and the plasmid together. So what we did was we took these fragments of the insulin gene, these fragments that contain the insulin gene, and we took these cut plasmids, we mixed them together, and some of them are going to stick together just by chance. This is the combination that we want. Then we're going to use ligase, and it's going to come in and it's going to seal the pieces of DNA together. And then we will take these plasmids and stick them inside of E. coli. Now we only need 1 E. coli to be able to work with this system. So if we're having a really difficult time getting this to work, we only need it to get to work once. We only need this to work one time, because once you have one bacterium and then it's confirmed, you can breed that bacterium up and get millions overnight. And of course, by the time you have it confirmed, you'll already have a colony. So this is how this recombinant DNA technology works. This is how the first product was basically created. So like I said, we did have to slightly modify the human insulin gene, but once we get this inside of E. coli and we get them producing human insulin, they will continue to produce human insulin for us. And there's other challenges we will face in biotechnology, such as plasmid curing, where the plasmid might get lost by accident, and then that particular E. coli says, well, hey, I don't have to make insulin anymore, so I can spend that energy just making more of myself. And as you can imagine, that is a problem in modern biotechnology, and there's other things along those lines. But the key thing here is once you have a bacterium that can produce human insulin, you can keep breeding that bacterium forever and keep making human insulin. So this is one of the wonderful things about biotechnology is once you have the prototype, you have a factory to make more. So CRISPR-Cas9 is one of the most important accidental discoveries made in the past couple of decades. So what this system does is it enables us to go into a nucleotide sequence, such as DNA, and make very, very small changes, make very small edits, such as changing just one letter of the DNA. And this system originated as an antiviral defense system in bacteria. So bacteria want to protect themselves from viruses, so they use CRISPR in order to do that. And scientists have adopted this as a tool to edit, delete, or replace DNA at very specific locations in a target cell. So if there is a G in a particular location and I want it to be a T, I can use CRISPR-Cas9 to go in and do that. Um, Perhaps we have a situation, like I talked about in class, where we have a pair of rabbits on a starship and we are wanting to settle another planet. We can use CRISPR to introduce genetic diversity, to remove negative genetic mutations, and to edit the rabbit DNA for our purposes. So two of the components used for this, the two major components, are we have a guide RNA, which is going to find the sequence on the DNA that we want to edit. So this guide RNA is like hitting Control F and typing in whatever sequence it is that we want to find. And then the Cas9 protein will cut both strands of the targeted DNA. So the guide RNA will find the piece of DNA that we want to edit, and the Cas9 protein will cut that piece of DNA. Now, the interesting thing about DNA is that it will try to repair itself. So once the DNA is cut in a very specific location by Cas9, the cut ends may be rejoined by DNA repair mechanisms that might introduce a random error. So this might knock out the gene, making it where the gene no longer has a function, which would allow us to figure out what the function is. So a lot of times we do that in biology where we will knock out a gene and then figure out what goes wrong, and that helps us to determine what was the function of that gene. There's been an analogy that it's like biologists take a bunch of TVs, have them roll down the stairs, and then once they figure out which part is broken, they can retroactively figure out what was the function of that part. So biologists, we like to break things and say, oh, well, apparently that's what it did. So a complementary piece of DNA with the desired repair sequence may correct a mutation, restoring the function. So this is where CRISPR-Cas9 technology gets really interesting. So we can take a piece of DNA and bring that piece of DNA to replace the part that we cut, and that lets us make the edits that it is that we're trying to make. And by using this technology, we can introduce very specific deletions or insertions or base changes. So this is where we can say, well, there's a G here, we wanted it to be a T, so we can go ahead and go in and make that change. And in the case of the pair of rabbits to settle a new planet very far away from Earth, if we want to introduce genetic diversity, say to help their immune systems or to help them to adapt to a new environment, we can say, well, these rabbits have an A in this location. Let's introduce T's, G's, C's, and create that variation so that we make sure we have genetic diversity within this pair of rabbits. And if you're watching this video without having seen my previous lecture, I do apologize for the missing context here, but hopefully you get the general idea. CRISPR-Cas9 can be used to make very small edits to a piece of DNA. So here we have a diagram of the CRISPR-Cas9 gene editing system. So we have the guide RNA, we have the Cas9 protein here, and then we have a matching genomic sequence. So here's a piece of DNA, and this guide RNA found the matching sequence. So now we're going to go in and cut that matching sequence. We might delete a gene, and we might insert a gene. So it depends on how we want to modify this particular piece of DNA. Here is a TED Ed talk. You can go ahead and watch this video on your own time. I definitely recommend watching it, maybe twice, and then it'll really explain to you how CRISPR works. So now we are on to the second part of the lecture. If you took this lecture with me in person, this is about where we left off. So we have another critical thinking exercise. And yes, I love to have my critical thinking exercises be out of the box, science fiction references, and things that go a little bit beyond the edge of what we know with our current scientific understanding. So Renee Holland is a wildlife biologist. She was on the show Finding Bigfoot, and she is described as being a skeptical scientist. So she had a term that she used in one of their episodes, and I wasn't able to track down the actual clip, so I do have to paraphrase it, and I'm sorry about that because it was a really brilliant thing that she said. But I remember watching this, I believe, in high school, and she said, as a scientist, it's not about believing or not believing, it's about evaluating the evidence. So she said that people often ask her, do you believe in Bigfoot? And her answer basically is, as a scientist, it's not about do I believe in Bigfoot or do I not believe in Bigfoot? It's about what evidence do you have for Bigfoot? So I really liked this, how it separated the question from whatever our personal beliefs are and just said, what is the evidence that you can lay out on the table right in front of you? So do we have pieces of DNA from Bigfoot? Do we have footprints that are supposed to be from Bigfoot? Do we have eyewitness accounts? And ultimately, do we have a body or a live Bigfoot that's been captured? So this is the interesting thing about questions like these. You have a lot of people who say, I saw Bigfoot, and then you also have a lot of questions that would make us skeptical, like, hey, why do we never seem to find a body? And so I really like Renee Holland's approach. It's not about do I believe or do I not believe, it's about what evidence do you have. And so I thought this was a very intelligent and very wise approach to questions like this topic. So that opens up the question, what is personal incredulity? And I'm glad you asked. So personal incredulity is another logical fallacy that is important to remember. Personal incredulity means you look at a particular topic and say, well, that idea is silly. I think it's silly. I think it's ridiculous. So I'm not going to believe it. So I think the idea of Bigfoot is silly. So I'm not going to believe that. And of course, whether or not you think an idea is silly or not has nothing to do with the logic of the argument, and it has nothing to do with whether or not Bigfoot actually exists. So this is what personal incredulity would be. On the other hand, I think there is a logical fallacy that we could probably call personal credulity. Um, I really, really want to believe in Bigfoot, therefore Bigfoot must be real. Or it's true because I believe it, or I feel that it's true. Um, Perhaps there's another term for that fallacy. However, I think calling that personal credulity, even though I tried to search for that and couldn't find anything, I think that would definitely be an appropriate term. So in science, it's not about do you believe in something or do you not believe in something? It's about what evidence do you have? And can you separate this idea from yourself and say, it's not about do I want to believe this or not want to believe this, just what is the evidence on the table? And the evidence on the table might not be strong enough to prove it, and you might want to believe it, so maybe you go look for more evidence. So, are human beings who are scientists totally without bias? No, of course not. However, you can separate yourself from ideas and say, what is the evidence? Not do I believe this or do I not believe this, but what is this? What is the evidence, and would this persuade an impartial third party? So the second part of this critical thinking exercise is if Galileo had been wrong, would it have been okay to persecute him? So a lot of people have some misunderstandings about what happened during the time of Galileo. This had nothing to do with anybody thinking the Earth was flat or not flat. People at this time knew that the Earth was round. People have known that the Earth was round for thousands of years. So people in the Middle Ages did not think the Earth was flat. That's a common myth. During the time of Galileo, Galileo was arguing that the Earth went around the sun rather than the sun going around the Earth. So he was arguing for a Copernican view of astronomy. And his research, believe it or not, was actually funded by a friend of his, the Pope. So he and the Pope were actually friends. Galileo had some ideas that were considered unconventional at the time. And at the time, people had a lot of what they thought were good scientific reasons to think that the Earth was stationary. And a lot of this had to do with, well, if the Earth were going around the sun, wouldn't we feel the motion? Wouldn't there be really strong winds? So they had a lot of what they thought of as good scientific reasons to think that it was actually the sun going around the Earth. Ultimately, they turned out to be wrong, of course, but Galileo came along, put forward his ideas. It was actually the Pope funding his research, like I said, and his ideas made it pass, went through the process. They passed the censors, and they said, "Yeah, go ahead and publish your ideas." This really wasn't an issue. But then what happened was there were other scientists who said, what Galileo is teaching contradicts what we're teaching. And we're making, a lot of money teaching the standard model. And so they went to the Pope and wanted to have their ideas canonized as official Catholic doctrine. Galileo came along and he created this character called Simplicio in a fictional in a fictional story that had a dialogue between several different people. Now, Simplicio, even if you don't speak Italian, you can imagine that name was not flattering. Simplicio had a very big hat, and he put the arguments of the Pope into the mouth of Simplicio, the person who was the fool in his story. And after this is why Galileo was put on trial. It probably had very little to do with his science in retrospect. The main thing was he was stirring the pot, which, he should not have been put on trial for that, of course. I think you should be able to criticize the Pope. I do it often. But this had a lot to do with politics, a lot to do with the self-esteem of the Pope, and I would say the pride of the Pope. And it had very little to do with the actual science at hand. True, the science at hand, is what started this whole thing, but a lot of this really had to do with egos and a lack of tact. Sadly, Galileo was put under house arrest as a result of this. But I would look at this and say, this was not a case of science versus religion. This was a case of an individual versus the collective. Does an individual have the right to stand up and say, I disagree with what the majority thinks? I disagree with what is considered the science of the time. So does an individual have the right to disagree even if they are wrong? So rather than seeing this, like I said, as science versus religion, I see this as an individual versus a collective, or an individual versus the establishment being allowed to stand up and say, I disagree. So one thing we've definitely learned from this scenario, hopefully, is how important it is to have academic freedom, how important it is for people in universities and in academia to be able to be wrong, to be able to have different ideas, to challenge established narratives, and to say things that, hey, might even be politically incorrect. So, if you want to learn more about the trial of Galileo, I definitely recommend the book The Genesis of Science, which is from a historian associated with Cambridge and Oxford. And he goes into a lot of detail of science during the Middle Ages, and towards the end, the book culminates in the trial of Galileo. So here on our next slide, we're going to be learning about polymerase chain reaction, or PCR. Now, the way a lot of this is described on here, I think it's very wordy, but we're going to go ahead and try to break this down. So PCR can be used to rapidly amplify specific DNA sequences to obtain millions of copies. So you can almost think of PCR like a Xerox machine for DNA. So a specific sequence of DNA can be amplified from within a large amount of genomic or contaminating DNA. So we have a whole bunch of DNA, but we want to amplify one specific sequence. And part of that, an example of that might be when you go and you get a COVID swab and they put it in the test tube and they send it off to a lab and it's tested by a guy like me. What happens is that even though you have lots of pieces of human DNA mixed in there, whatever bacteria you have growing in your mouth, maybe pieces of whatever you had for lunch, all of those different pieces of DNA are mixed in there. But we're going to use primers to specifically target just the DNA from the SARS-CoV-2 virus. So the DNA to be amplified, primers, nucleotides, and heat-stable DNA polymerase are combined. So these are all put together in a test tube. So you have the DNA, including the DNA that we want to amplify. We have primers, which are going to find specific sequences of DNA. You have individual little nucleotides, and as you can imagine, those will be important because if we want to build a chain of DNA, we need the little nucleotides, the little A's, G's, C's, and T's floating around in order for there to be some raw material to amplify the DNA. And we also need a heat-stable DNA polymerase. So the DNA polymerase is going to say, okay, here is a piece of DNA, here's the primer. It's going to come to the primer and then make copies of the DNA that primer binds to. And we will see diagrams of a lot of this here shortly. So repeated heating and cooling cycles allow for rapid amplification of a sequence of DNA defined by the primers. So what happens with that is you have DNA, of course, is a double helix. So you have both pieces of DNA together, and when you heat it up, they separate. So as hydrogen bonds break, and then a primer comes in, binds to a piece of DNA after we cool this down a bit, and then the polymerase will come and make a copy of that DNA. So we started with one set of DNA, they were broken apart, and now we have two copies of this DNA. And then we'll add heat again, so they'll break apart, and it'll cool down, a primer will come in, polymerase will come in, and the whole process will start all over again. So you start with one piece of DNA, 1 becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16. And it's the primers, let's underline that right here, the primers that specify the sequence to be amplified. So here we have some applications of PCR. This can be used for crime scene investigations, so maybe there's some saliva on a cigarette or a drop of blood or a hair follicle. Any piece of tissue that came off of a human body that would contain DNA can be used for this process. So amplifying DNA of a pathogenic microorganisms to aid in diagnosis of infectious diseases. This is done a lot in clinical labs, of course, and the RNA of SARS-CoV-2 is one major example of that. And we can amplify DNA prior to sequencing. So say there is a sample of DNA, and we want to sequence that sample using something like the Sanger method. Well, we need to have enough DNA in order to do that, so we might use PCR to make millions of copies of that one piece of DNA. So with the PCR for the detection of the RNA of SARS-CoV-2, the way that works is you have a reverse transcriptase So it takes a piece of RNA and reverse transcribes it into DNA. So we're going backwards. And then that piece of DNA is copied using the polymerase chain reaction. So, polymerase chain reaction, like I said, it's basically a Xerox machine for DNA. We can use this for forensic samples to take the piece of DNA and make many, many copies of it, and then use that for DNA profiling. We can make copies of DNA to detect pathogenic microorganisms. Perhaps that's SARS-CoV-2, or perhaps it's chlamydia and gonorrhea, which is a popular test in clinical labs. You can amplify DNA prior to sequencing. So, hey, maybe we found a hair that we thought was from Bigfoot, and we want to amplify the DNA. We can use PCR for that to make lots and lots of copies, and then we can find out using other methods later on, like, hey, is this an unknown species of ape, or is this just a piece of hair from someone's dog? So, and like I said, modification of PCR can be used to take RNA, reverse transcribe it to DNA, and then we can test people for COVID. So here's a basic diagram of how a polymerase chain reaction works. So you have a piece of DNA, you heat it up, the two strands break apart from each other, and then the primers will bind to the DNA. And then DNA polymerase will come along and it will copy the piece of DNA onto the reverse complement strand. And so we went from having one piece of DNA to two, and then we will heat it up again, and this will break the different pieces apart from each other because it'll break the hydrogen bonds between the two ladders. And when we cool it down again, then the primers can come bind again, and DNA polymerase can come along and copy the DNA. So we went from 1 to 2 to 4. So this is a very, very interesting setup. If you ever look into the history of how polymerase chain reaction was developed, you definitely have a lot of funny moments, and I highly recommend it. So that's definitely something to check out. So identifying the source of DNA. So this is DNA profiling, otherwise known as DNA fingerprinting. So we can use this to identify suspects in criminal investigations. So we found a piece of DNA at the crime scene, and we want to know, did this person commit the crime? Now I've talked to students before, and I asked, well, hey, here's a scenario. You find a woman murdered in her bedroom, and you find her husband's DNA on her. Does that prove that the husband committed the murder? Not necessarily. So you have to understand what DNA is and understand it in context. And also understand that people leave DNA behind basically wherever they go. So understanding the role of DNA in criminal investigations and in law is a very important thing. So some of you might be majoring in something like political science, or you might be majoring in pre-law, and you might say, hey, why do I have to take this biology class? This is stupid. But this is a major reason. When you go and you create laws, or you have to weigh the costs and the benefits of a new type of DNA technology, you really have to understand how this works. You have to understand how DNA works if you're going to be on a jury, if you're going to be making laws, if you're going to be arguing in court. DNA is not magic. It's not something that should only be understood by scientists. It's something that everybody should understand. So identifying the suspects, if we find a piece of DNA at a crime scene, paternal determination or paternity determination, so we want to know who's the daddy of the baby. Identifying human remains after disasters. This is very sad, but it's necessary. If we find a body, we have to know who that body belonged to. We want to be able to give closure to the family and hopefully make the best we can of a bad situation. So unknown DNA from a crime scene or a disaster site if compared to known samples. So if we don't know what the body is, or who had the cigarette butt, or whatever it is, we can use the DNA that we find at the site to figure out who was there, figure out where the DNA came from, and put things together from there. So, identifying the source of DNA, the regions of DNA that are compared are the areas that are hypervariable. That means they differ a lot among individuals. So, we talked before about how if you have any two people in the world, so if you have somebody from Scandinavia, tall, blonde hair, blue eyes, and you have somebody who's native of South Africa, very dark skin complexion, very dark eyes. These two people might superficially look very different from one another, but their DNA is almost identical. The DNA of any two human beings in the world is almost the same. And I've said before that if you were to have somebody from Northern Europe, South Africa, East Asia, and somebody from the Americas, and you had a space alien look at the DNA from these four men, the space alien wouldn't just say these four men are brothers. The space alien would say these four men are clones. They're almost identical to one another. So, given that any two human beings on Earth, our DNA is almost identical, how can we tell the DNA apart by looking at DNA from, say, a crime scene? Well, fortunately, there are hypervariable regions. So these are very, very small regions of the DNA where any two individuals tend to be very different from one another. And one classic example of this is areas where you have short tandem repeats. So short tandem repeats are 2 to 5 nucleotide base pairs, so 2 to 5 letters long, that repeat over and over and over again. So it might say ATC, 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 ATC over and over again. But the number of repeats is where it's going to vary. So maybe I have 20 repeats, but Joe has 30 and Tom has 40. That means this is an area where you can look at our DNA. There's a lot of variation, and you can use this to figure out using DNA fingerprinting, to figure out who left behind this piece of DNA. So, what does all of this mean? So STR regions vary in length depending on the number of repeats. So in a particular STR region, perhaps I have 20 repeats and perhaps Fred has 30 repeats. So DNA samples are collected and STR regions are amplified using PCR. So PCR will target these regions that contain the STR regions. And the amplified DNA is then subjected to electrophoresis, which separates DNA fragments by size. So the DNA fragments will go through this gel, and if you were to look at this gel on a very small scale, it would almost look like a whole bunch of little ropes connected to each other. So a piece of DNA traveling through this, because it's traveling from the negative end to the positive end, if it's a longer piece of DNA, it's going to get tangled up a little bit more often, versus a smaller piece of DNA is going to be able to go through without getting tangled as often. So the shorter fragments are going to move faster than the longer fragments. And the distribution patterns of amplified DNA fragments are compared. It kind of looks like a barcode by the time you're done. So if an unknown sample pattern matches that from a known individual, then the unknown sample is presumed to have come from that individual. Now we want to look at more than just one STR region, because say you have a 1 in 100 chance of having the same STR pattern for one region as another person. So say it's me and Fred. Well, okay, that's still 1 in 100 times that our DNA could match. You know, is that really something that's impressive enough to be used in a court of law? I would say no. So now say we use 2 regions and say, for the sake of argument, it's, 1 in 100 chance of having the same number of repeats in those regions. Okay, so now it's 1 in 10,000 that we had, the same pattern. So as you can imagine, you don't want to have just a few regions that are the same. you want the odds of these two DNA fingerprints matching to be astronomical. So you want to be able to go to that jury if you're a prosecutor and say it is a 1 in 100 billion chance that this individual who is on trial, that his DNA would have matched that found on the cigarette butt by chance. Now you start having some stronger evidence. And now the defense has to say, well, the cigarette butt came from my client, but he was at the crime scene earlier that day. He wasn't there when the murder was committed. So DNA itself doesn't, just by itself, it doesn't prove that somebody committed a crime. You do still have to rationally interpret the evidence. So this is why understanding DNA technology is very important. This is why we have to understand what DNA evidence means. So here we have a chart, and we've already pretty much explained this process. So we cut up pieces of DNA and we used polymerase chain reaction to amplify those regions of interest. Now we're going to take our DNA samples and put them through a gel. So we have this unknown sample, sample 1. Then we have sample 2 and sample 3. So these two individuals are suspects. Now the fact that, the profile indicates that one seems to be a woman and the other seems to be a man, well, you think this would be an easy one to figure out, but we're still going to do the DNA fingerprinting. So we could see this DNA profile right here. We can see this DNA profile right here. And we can see how sample 1 matches sample 3. So if I go through this, it looks like we have a match. Now, say this one were really skinny and this one was fatter, then it would say, okay, these two do not match. But would that necessarily be the end of the story? Maybe, maybe not. I think it depends on what other evidence do you have. Do we want to run samples again? Did DNA break down? So there's a lot of things you would have to take into consideration. But overall, it looks like we have a match between what we found in this cigarette butt and this gentleman right here. So he might have to explain, how did his DNA get to the crime scene? Then again, we also have presumption of innocence in this country. So, if he's innocent, hopefully that comes out. Hopefully the truth comes out. If he's guilty, hopefully the truth comes out. So a transgenic organism, this is another important term, is an organism that has been genetically engineered to carry one or more foreign genes from another species, also known as a genetically modified organism. So transgenic bacteria and yeast have been developed to produce the following. So we already talked about insulin. That is a classic example in biotechnology and what started off the modern biotech revolution. There's also human growth hormone, erythropoietin, tissue plasminogen activator, and factor VIII, which is a blood clotting factor needed by many hemophiliacs. And we also use this to create new vaccines. So bringing this back around to the original critical thinking exercise, Dr. Flox could point out that humans have used genetic engineering to create a lot of different important products. So we've used genetic engineering to create new vaccines to help make our lives better, to create artificial insulin, and to create a lot of other products. So this goes to show that while any particular technology could be used for evil, hopefully we are choosing to use it for good, and hopefully we decide that genetic engineering is a positive technology, and we could have a future like the people on the fictional Denobula, where this technology helps to make the world a better place, and where individual rights and dignity are upheld and respected. So here's a picture of a worker at a vat, and the caption says transgenic bacteria and yeast can produce vaccines. Pretty straightforward. So right here inside of this vat, you can't tell just by looking at it what's actually being produced. I'm presuming this was stock footage. I'm presuming that we have no idea what is being made inside of this vat. However, we could grow transgenic bacteria or transgenic yeast in a vat like this, and perhaps these were transgenic organisms used to create a new vaccine against one of the many, many diseases that still plague humanity. So, as the caption reads, transgenic plants have been modified to express the following. So we have increased resistance to freezing. Cool. Longer shelf life. This definitely makes me think of the flavor savor tomato. Increased vitamin A. Sounds a lot like the Golden Rice Project. Um, Vitamin A is the most important vitamin in terms of malnourishment around the world. There's a lot of people who go blind from having a lack of vitamin A. So that is a major problem that the Golden Rice Project was trying to solve. Um, Now, in the last few decades, starvation level poverty around the world has gone way down. So if you want to look at something positive in the world, the rates of poverty are dropping like a stone. So look at the rates of poverty in 1970 and then look at the rates of poverty today and look at how that has changed over the course of the last few decades. So we also have edible vaccines, human proteins such as albumin, increased resistance to pests, resistance to herbicides, and drought resistance. So we are doing a lot of things in order to make better crops, make plants that we can use to help feed more people and take care of the people in this world. So I looked up this example that the textbook gave us. What we have here on the left, and I presume they mean our left, is a leaf from a poplar tree, and this is a normal poplar tree, versus a poplar tree that was genetically engineered for its leaves to grow larger. Um, I'm not sure what the purpose was for this particular project. The textbook didn't give us a lot of information. We could look into it further if we really want to find out more. However, this is an interesting proof of concept because we might need plants to be able to do a job that natural plants are unable to do. Maybe we need a particular product that we're just not getting from the plants that we have. Or perhaps perhaps we want to conserve the forest and maybe there's some kind of pest that's destroying the trees. We might need to say, hey, we should have a genetically altered version of this tree that is able to resist the pests. And you might say, hey, at what point are we playing God with nature? Then again, if we are, people asked a philosophical question, here's a philosophical answer. If we're to steward the planet, then perhaps genetic engineering might be the best way to help to solve certain environmental problems. So again, I don't know what the exact purpose was for this particular project. Obviously, as a biologist, I cannot know everything. Um, but it does give you a proof of concept that there's a lot that can be done with genetic engineering technology. So when it comes to genetically engineering animals, we have some bigger challenges. So if I want to genetically engineer corn and I wanted some seeds of corn, I go out into the field and I pluck an ear of corn. In order to obtain eggs from a goat, that might be a little bit trickier. So it's more difficult to introduce foreign DNA into animal cells, which is also mentioned here. With plants, you can use something called a gene gun, and I'm sure there's lots of other methods that we've developed in recent years. But for animals, we have this microinjection of DNA into an egg. Here's one possibility. Or we might use CRISPR-Cas9 to introduce a desired piece of DNA. And this might go beyond just editing a few nucleotides here and there. Perhaps we want to make a larger change using CRISPR-Cas9. Don't assume that CRISPR-Cas9 is only limited to modifying a few bases here and there. So cloning animals from a single cell or a group of cells is more difficult. So we talked before about using a embryo at the 8-cell stage, breaking apart those cells from each other, and then using those to develop 8 different individuals. So that is a possibility of something we can do. And perhaps we can modify, those embryos at that stage. So we first must introduce a gene of interest into the fertilized eggs, which are then implanted into a female for gestation. So you need to implant the fertilized egg into a receptive female who that can grow, this particular embryo. Whereas if I genetically modify a kernel of corn, I put it in the dirt. So it's a lot easier to plant that seed than to plant a fertilized embryo. So only a few mature eggs are available at a time with animals, which limits the number of transgenic animals that can be produced at any one time. And of course, this will no doubt be easier with some species of animals than others. Salmon, of course, produce quite a few eggs at a time, and they just will dump them right out into the water. So on a technical level, it'll probably be a lot easier to genetically modify salmon than something like a goat. But it can be done, and it has been done. So there has been success with transgenic animals. I know it says some success, but we do genetically modify mice in laboratories all the time. So, I mean, it's not like the difficulties are insurmountable. You just have to work a little harder. So, successes, we have transgenic pigs. So, a gene from a roundworm was introduced. The pigs produce meat with fewer unhealthy fats and healthier omega-3 fatty acids. I'm going to give an opinion here and say, you know what, just go have some chicken, go have some steak. It will be fine. Um, I don't think that all of this is really necessary. That's my opinion on the matter. And go take some omega-3 fatty acids while you're at it. Um, Transgenic mice with the human ACE2 angiotensin converting enzyme 2 gene was introduced to the mice. These same mice can now be infected with the SARS-CoV-2, providing a model to study the disease. I've got some mixed feelings about that. I think I'll just leave it at that. There could be good and there could be bad with this. There's also gene farming, and notice farming spelled with a PH, which is introducing human genes into dairy animals in such a way that the human protein is produced in the animal's dairy milk. So we will see that here on the next slide. So here we have gene farming. So we have a goat that has presumably been genetically modified to produce some kind of human gene. Now, if you go and you do a little Google search, you can learn about spider goats, which were a thing at one point. I think the contract was actually bought out by the military, where they were using goat milk to produce a protein in spider silk. And then they were able to take this protein and use it to make silk string. So the military said, hey, spider's milk or spider silk, sorry, is very, very strong. We like this technology. We would like to buy it. So at least that's the version of the story I heard. I remember seeing documentaries about the spider goats, and last I heard that's what's happened with them. So here we have the producing human proteins in transgenic animals. So you have a plasmid, just like with a bacteria. We're going to put it inside of a fertilized goat ovum, or perhaps we put it in an unfertilized one to start with. And then fertilize it afterwards. I mean, not sure if there'd be a difference one way or the other. So then we put it inside of the mother goat, and then there's the transgenic offspring, and this offspring produces this milk with the protein of interest. So gene therapy, is this the hope for the future? So this is where things start getting a little controversial and where I really like the perspective of somebody like Dr. Flox. So gene therapy is the introduction of human genes into human cells to treat or eliminate a disease. Some obstacles are that it's difficult to introduce genes into the right cells where the genes would normally be expressed. So we want to make sure that if there's some kind of gene that is broken and we want to introduce a functional version of that gene, we want to make sure that the functional version of that gene is sent to the correct cells so that we can have the effect that we're looking for. So we need effective means of delivering genes into all somatic cells, either all of them, or into the specific tissues or cell types, that are affected. And we want to have corrective genes that can be introduced into. This is where things start getting a little controversial, or where ethical questions start to get raised. Can corrective genes be introduced into reproductive cells, so sperm and egg, to stop the passing of defective genes to offspring? So at what point in gene therapy, if we have it where there's a congenital form of blindness, at what point do we start to correct errors in the germline? At what point do we say we are confident enough where we can go into the cells that are going to produce sperm or egg, or go into the sperm or eggs themselves, or go into an embryo and say we are going to use CRISPR or use some other technology to correct the defective genes? And somebody might say, like, hey, is that something we should be messing with? And so that's where you have to say, what is the cost benefit? What is the risk reward assessment? And at some point, I do think that it would be a good idea. I'd be a little skeptical to say the technology is totally there yet. But at some point, I would like to see a future where congenital disorders can be corrected, where we can go in, without destroying a human embryo, without raising a lot of the ethical concerns we might have, and be able to correct errors in our DNA, so that we can have a world where we have a lot less to worry about in terms of genetic diseases. Now, I do obviously have the same fears that many people would have, that this would be turned towards eugenics, turned towards, oh, we want our baby to have purple eyes because purple eyes are in this year, or having it where natural genetic variation, such as different forms of the autism spectrum, are unnecessarily eliminated, or having it where, like, hey, here's a society where we want everybody to be the same. Because at what point do you have that line between what is variation and what is defect? So being able to manage all of that and say, okay, where do we draw the lines? Where do we, make those decisions? While also still maintaining the rights of the individual and the dignity of the individual. Those are all kinds of questions that are raised with gene therapy. On the other hand, there's also the question of doing nothing. So any technology comes with a risk, but doing nothing also comes with a risk. And I think that's where, if we step back into the fictional world of 2170, why I think that it's important to hear from people with different perspectives, with different cultural backgrounds. And maybe that's from the Denobulans, who have a different history from our own and who have a positive experience with genetic engineering, and perhaps they can help to guide us to help make wise decisions as humanity pivots towards a more positive future. All right, so now let's talk about vectors that are used to transfer genes to a human cell. So what are vectors? Vectors are basically the delivery mechanism to bring the DNA into the cell that we're targeting. So you can almost think of a vector almost like a taxi. So you take the little piece of DNA, put it inside of the vector, and it will take it to the cell that we're trying to take it to, and bring the DNA inside of the cell. So one classic vector are viral vectors. So we basically will build a viral capsid, and we will, instead of putting viral DNA or RNA inside of it, we'll put the gene, the human gene that we want to put inside of that cell. So perhaps there's a particular gene that's broken inside of those human cells, and we want to have a healthy version of that gene introduced into those cells. So you can use a viral vector, and this can be done either ex vivo or in vivo. So ex vivo means outside of life, and in vivo means inside of life or inside of the body. So with ex vivo gene therapy, viruses carrying the human gene of interest are introduced into cells that have been removed from the body. The infected cells are then returned to the patient. So for example, you might take bone marrow cells, take them out of the patient, introduce the viral vector, and that will contain the gene of interest that you're wanting to insert into these human cells. This can then be followed up by taking those cells and reintroducing them into the patient. So in vivo is when we take the viruses and we directly inject them into the patient. So if it's bone marrow again, you would inject these viral vectors directly into the bone marrow, and then they will introduce the gene of interest into those cells. You can also use lysosomes, which are small fatty spheres surrounding the gene, and these can connect to the cells of interest and basically connect with that cell and fuse with it like a small bubble connecting to a larger bubble. And then after it fuses with the cell, it can introduce the DNA of interest. So we can also use naked DNA. So that would be basically taking a piece of DNA and doing something to the cell so that the piece of DNA can cross through the cell membrane. And we could do that by a number of different methods. So it says here all of these methods are experimental. So this is ongoing. A lot of research is being done in this area. Sadly, because of tragedy in the past, there has been a history of gene therapy not working as expected, and that put a bad taste in the eye of the public. Of course, there's the flip side of that. You know, how many people are dying from genetic diseases needlessly when a little bit of admittedly risky research could be done to help to save a lot of lives. So here is a diagram showing how a viral vector works. So normal gene is put inside of a viral vector, and by normal gene we mean a healthy gene, and then this is delivered into the cells of the individual. And in this case it's ex vivo, so we're introducing this into a cell that was taken from the patient and then re-injecting this into the patient. So here's an example of some advances that have been made in gene therapy. So severe combined immunodeficiency syndrome, or SCID. This is an inherited disorder where individuals lack an enzyme, adenosine deaminase, ADA, and this causes a deficiency of B and T cells. So as you can understand, those are very, very important to the human immune system, as we'll be learning about pretty soon in terms of immunology. Without B and T cells and the role they play, you're not going to be able to make antibodies. So children are highly susceptible to infections. Ex vivo gene therapy attempts to introduce the gene for ADA into the bone marrow stem cells. So bone marrow stem cells are harvested, infected with the viral vector containing the normal gene. Again, this viral vector contains the normal gene. It does not contain any viral DNA or RNA. So we're cheating the system in a way. It's then returned to the bone marrow. So these cells, after they are infected with the viral vector containing the normal gene, they are returned to the bone marrow. And hopefully this means that this individual can then go ahead and make B and T cells. And it says here there have been some encouraging results. It would definitely be interesting to read more about this. So here's some more advances that have been made. So there's mutations in the RPE65 gene, or retinal pigment epithelium gene, and this can lead to blindness. Viral vectors with the normal version of the gene are injected directly into the eye. So this is in vivo gene therapy. So in vivo, it means they are injected directly into the patient. And this is the first FDA-approved gene therapy, Luxturna, which was approved in 2017. So anytime we see that there's a brand name going on with this stuff, that's pretty encouraging. It shows that the technology has come a long way. I remember when I was in school studying a lot of this stuff, and I graduated in 2015, we were learning about studies like this to help to heal people of blindness, but they were still very, very early, and um, because of a lot of the tragic history of gene therapy not going as expected, the science was developing very slowly. So this is the end of our slideshow for now. I hope you guys enjoyed this. If you are interested in learning more about biotechnology, definitely talk to me. This is what I majored in when I was at RIT, so it's encouraging to see a lot of the advancements that have been made and a lot of the progress that has been made in many of these fields. And of course, no one person can know everything. So if there is a particular area of biotechnology that interests you, such as gene therapy or genetic engineering of plants, there's a whole world out there. There is so much to learn, and you can spend an entire career specializing in any one of these areas. And of course, there's a lot of very intelligent, very friendly researchers here at RIT, which is something that really makes RIT very special. We have professors who are willing to teach, but are also very, very happy to talk to students and very happy to share about their research. So if you want me to make any introductions, definitely talk to me, and I will see what I can do.